This is a presentation that was held at the 12th Haploid Markers meeting in Hungary, 2023, with the title Advancing Forensic SNP Typing and Insights from an Interlaboratory Study of the Force Panel. So here we wanted to present some results from a study that has been performed during the last year regarding SNP marker analysis in forensics, and specifically a forensic SNP panel called the Force panel in an interlaboratory study fashion. So as you can see, a lot of labs participated in this study, 15 different labs around the world. And this presentation, we will talk a little bit about SNP typing and SNP markers for forensic applications, and specifically the use and the test of a specific set of SNP markers, the Force panel, analyzed with different assays. So just to introduce, when it comes to SNP markers, normally another type of DNA markers called short tandem repeats, STR markers, are the ones that are commonly used in forensic applications. However, SNP markers have also been around for many, many years without being given that much attention for different reasons. There are some advantages with SNP markers to be used for both forensic, kinship, and missing person analysis. One, for example, is that these type of markers are suitable for the analysis using high-throughput sequencing technologies like next-generation sequencing or massive parallel sequencing. Another advantage is that these type of markers, since they are only one nucleotide long or short, it makes it possible to analyze very, very short amplicons or fragments, which are very useful in forensic settings when you may have degraded DNA or DNA of bad quality. Another advantage is that there are millions of SNPs to choose from in the human genome. And for kinship and relationship testing, the low mutation rate compared to STR markers is beneficial when it comes to establishing and predicting relationships that are very, very close, like parent and child. And the SNP markers are also relatively easy to interpret for the genotype calling when it comes to in comparison to STR markers, which makes things a little bit easier as well. However, as I said before, SNP markers have been used, although in very limited scale in forensics, and we think there is a need for standardization of forensic SNP markers in order to improve and be able to use it more broadly than has been so far. However, what we do see is that many different forensic laboratories have different preferences and experience when it comes to, for example, library prep methods, how to analyze, for example, SNP markers, which techniques and technologies. Also, different laboratories have different sequencing instruments. Different laboratories have different bioinformatic tools and experience and know-how. So the question was, when we started the project before this interlab study, was to see if it is possible to customize the complete workflow for a specific large or medium-sized 5,000 SNP marker panel, going from DNA to genotype. So instead of establishing one method, we wanted to design a panel consisting of a set of markers and then test if these set of markers could be applied for very different assay types, depending on each lab's possibilities and preferences. So therefore, when we started the Force project a couple of years ago, 3 or 4 years ago, we wanted to select a recent number of markers to be able for various forensic purposes, like identity testing, kinship testing, but also predictions of ancestry and phenotypic characteristics like eye color, hair color, and prediction of skin color. What we wanted when we did this design or selected this marker were that we did not want to include any novel SNPs, meaning that we only wanted to include SNPs that sort of previously have been shown to work and that there are earlier information about these markers and well studied, for example, different populations and so forth. And the reason for this, for example, was to aid cross comparisons. For example, if one uses the force panel to establish a SNP profile from a crime scene sample and a reference sample, perhaps were established at another lab with another method, if we have the same sort of SNPs included in both assays, this would make comparison possible. But also during validation, it's very good to have the same SNPs, or at least some of the SNPs included in other assays for validation between different methods. So for example, looking at the markers that are included in the force, for example, the kinship SNPs, all of the kinship SNPs that we have included are also included in this in the major SNP chips, like the GSA chip, OmniExpress chip, etc. And we also, for example, the ancestor SNPs are SNPs that already have been developed and included in other panels like the Visage panel, the Precision ID ancestry panel, and QIAGEN's forensic signature prep panel. So a little bit more about the 4 SNPs. As I said before, there are roughly 5,500 SNPs in total, and the SNPs are for various applications like ancestry testing, and the ancestor SNPs, around 240 of those, can be used to predict biographical ancestry for unknown individuals. We have the phenotypic SNPs included for the prediction of eye color, hair color, and skin color, and these are SNPs that are already been developed by Manfred Kaiser and Susan Walls and colleagues, and they also have interpretation tools that can be used and applied. But the vast majority of the SNPs included are for kinship and identity testing. Almost, a little bit more than 4,000 SNPs are can be used for kinship prediction or identity testing. And using that amount of SNPs, you can sort of predict and establish relationships up to 5th degrees of relationship. So I would say very useful for most of the forensic and legal kinship questions that may arise. We also have some X-chromosomal SNPs for different relationships that have such a degree of relationship that can be separated by the use of the information from X-chromosomal inheritance patterns. We also do have around 800 Y SNPs included in the panel, and these can be used to predict the biogeographical ancestry on the paternal lineage, but it can also use for paternal lineage comparison. For example, if 2 male individuals would like to see if they share the same paternal ancestry. And the Y chromosomal SNPs have earlier been developed by Manfred Kaiser and Arvin Rolf in earlier studies. So as I said before, we did want to test if it was possible to design a panel and with the same set of markers you use different assays or technologies in order to establish the genotype for this set of SNPs. So currently there are 4 different assays available to establish SNP profiles using for this force marker set. One is called MyBates technology, and it's are built on a hybridization capture technique. I will go into these techniques a little bit more in the next slide. But there are also others like the QIAseq from QIAGEN, the AmpliSeq, which are built on the traditional PCR amplicon amplification technology or Thermo Fisher, and a similar one from integrated DNA technologies. If you want to read more about the MyBates implementation, it's in the original Force panel from 2021. And if you want to know a little bit more about the QIAseq implementation, there is a paper quite recently out in 2023 from Stadig et al., where they tested the QIAseq Force panel in more depth. So very briefly about the differences between the different technologies. So the MyBates implementation of the Force are built on a hybridization capture. So what it's built on is that you have your genomic DNA, which are fragmented, and adapters are ligated to each fragment. And then the fragment that contains the SNPs of interest are captured using probes that are designed to hybridize and capture the fragments that contains the SNP of interest, which means after the capturing step, you only have your fragments containing the SNPs of interest. You do a library amplification, and then you sequence all the fragments using like a MySeq or a NextSeq or an Ion S5 instrument. The QIAseq version is quite similar in many ways. So you have your genomic DNA, you fragment DNA, you add adapters, but you also add something called UMIs, which are unique molecular indices, which are unique DNA sequences that are tagged and unique for each individual template fragment. And this can be used in the bioinformatics afterwards to remove all PCR duplicates. The fragments that contains the SNPs of interest are then captured in the QIAseq. These are captured using a single primer extension approach, which means that there are one primer that anneals close or relatively close to the SNP of interest. And those fragments are then amplified, which means that the fragments that do not contain any SNPs are reduced in a manner that you in your library only have in principle the fragments that contains the SNP of interest. And then you do sequencing as you do with similar methods like the MySeq, NextSeq, or Ion S5. And the third variant is the PCR amplicon sequencing, and this is a traditional amplicon based that you have a forward and reverse primer for each SNP, and you amplify and there target and capture the fragments or the target of interest using PCR amplification, and you add adapters to those amplified regions, and then after another library amplification, you do sequencing as with the previous methods. So now to the Force interlab study. So what we wanted to see, as I've said before, is it possible to customize the complete workflow, which means that we wanted to see if each of the participating lab, based on their own preferences when it comes to choice of the assay type, when it comes to parameter setting for the sequencing, the number of samples to multiplex or sequence together, and things like that, could be result in useful outcome. So in total, 15 different labs participated, and the labs represent labs all over the world with, I would say, both less and also more experience from these type of analysis. So all 4 different assay that has been developed through the years for the Force panel were included. So there were MyBates, the Chi-seq, the AmpliSeq, and the XGen. And also there was a representation for different sequencing instruments like the MySeq, the NextSeqs, and also Ion S5 sequencing instrument. So each lab, they got 12 different DNA extracts representing both good quality samples like the Coriell reference DNA samples and also NIST reference samples. They also got the dilution series ranging from 30 picogram total input up to 10 nanogram total input, and also a number of bad quality samples like degraded DNA and the inhibited samples that were inhibited with humic acid. So each lab were then tasked to choose their assay, sequencing instrument, sequencing strategy, etc., based on their own choice and premises based on earlier experience, or if they didn't have an earlier experience, they just follow the protocol that exists for each of the different assays. We believe that doing an interlab study like this, instead of the alternative to give everyone exactly the same protocol, we believe that this makes it more realistic because different labs have different approaches. So it would be more useful to learn from the outcome from this study. However, of course, since all different labs have different settings and different variability when it comes to this, it makes it a little bit harder to compare the outcome in an ordered way. So just to briefly show the variability, so here you see each row here representing a lab and its sort of setup. So there were all, as I said before, all assays were included, different sequencing instruments, sequencing kit also, and also the cycles, the sequencing cycles and approach there differed between the different lab, and also the number of samples to be sequenced together. And of course, the number of samples sequenced together will have an effect on the coverage or the sort of the number of reads output per sample, which we will see later on in the results. So for consistency, all the genotype calling was done at one lab, and we used CLC genomic workbench for this. Of course, due to the fact that the different assay types have a little bit different chemistry and different characteristics, we could not use the same identical bioinformatics workflow for each assay type. Like for example, the MyBates, we could remove all PCR duplicates, and also for the Chi-seq when we use the UMIs. But for the AmpliSeq and XGen, since these are amplicon based, there are no way to remove PCR duplicates with these two types of methods. So that's where one of the major differences when it comes to bioinformatics. The genotype calling after the bioinformatics was done using in-house developed R script based on an Excel spreadsheet that Kim Andriagi developed a couple of years ago connected to the FORCE project. And the results that we will show are based on different thresholds for the genotype calling, like the coverage, the allele read frequency, and also the quality for the nucleotide sequencing quality, and also forward and reverse read ratio. So some results, we don't have the time to go into depth in this, but just to give an insight in how it worked. So this was a good quality sample, and what you see here is the autosomal SNPs, and for a Coriell sample, and the green color or the green bar represent correct and called genotypes, The white bar, or the white proportion of the bar, represent in this case no calls. So then the sum of the threshold were not met for the genotype calling. And for this, you also see a little bit dark coloring, a very, very small proportion, and these are incorrect genotypes, genotype errors like allele dropout or allele drop-in. But for a good quality sample like this, you see that for each of the different, let's say there were at least one or even multiple labs, or labs that did get in principle 100% call rate with 100% in principle correct genotypes. So this shows that all the different assay works at least on good quality samples. So overall, very good call rates and accuracies across assays. However, we did see some variations, which can be explained. For example, this MyBates 5 lab, they chose to sequence all the 12 samples combined in one sequencing run, also using a micro reagent for the sequencing. And of course, the consequence here is that the total read output from the sequencing is not in the same range as for the other, meaning that there is a higher probability, like we see here, that each SNP does not get as high coverage as needed to reach the 10x threshold, for example. We did have also another example that could be explained by poor or bad sequencing reagents quality, which also made the results not looking that good in terms of coverage. For the AmpliSeq, we also did see for some of the SNPs some consistent errors, genotype errors based on mapping, or based on the fact that there are complex region for the mapping, like you do see in these two examples. So there were both false inclusions of alleles and false exclusions alleles due to this. And these were consistently observed for a few SNPs. And since the AmpliSeq design was tested for the first time here, these are things that we find in this interlab study, which make it possible to also, of course, in the future exclude these SNPs for analysis due to complexity in the mapping. So even though we didn't see that much for good quality sample variation across, most of them have good call rates and high accuracy, we did see differences when we looked at the underlying data, especially when we looked at the coverage data. And this, of course, has the reason in the way the different labs chose to, for example, do the sequencing and things like that. And even though it did not affect the good quality sample, the coverage, of course, can have a major impact on the genotype calling for more for DNA samples of perhaps less high quality. It's not easy to compare across all the coverage across all different methods, because for the QIAseq and the MyBates, all the PCR duplicates have been removed. So these are the coverage after PCR duplicates removal. However, for the AmpliSeq, since you don't remove any duplicates here, it's not that easy to compare the coverage with the QIAseq and the MyBates. So this should be kept in mind. So what about looking at more perhaps forensic-alike samples and more problematic samples? This is the corresponding result with the coverage call rate, not the coverage, the call rate and the accuracy for a degraded sample. This sample has a mean or average fragment size of around 200 base pair. And what we did see was that a consistent bad result for the AmpliSeq design for this explicit sample, while the QIAseq and my baits worked fine. So the reason for the high proportion of no calls for the AmpliSeq was probably due to the fact that the AmpliSeq on average designed amplicons of sizes around 250 base pair long amplicons, which mean that some of these amplicons, or for some of the SNPs, or majority perhaps of the SNPs, had longer fragments than these 200 base pair that the DNA was fragmented to. The inhibited samples worked fine, and here we do see that it worked fine both for the QIAseq. The AmpliSeq also had representation of in principle complete profiles, as well as the XGen and the MyBaits. So what about dilution series? Of course, in forensic settings, it's also very important that you're able to produce and obtain SNP profiles from extraction or DNA samples that do not contain that much DNA. So for example, looking at 100 picogram total input, even though we do see a large variability across the different labs, we do see for the QIAseq and the AmpliSeq and MyBaits lab that produced sufficiently, or at least a high amount of calls with high accuracy. So it shows that for these assays, it's possible to obtain useful data. However, there are labs that, most probably based on their strategy and sequencing strategy, and for example, number of samples to multiplex, did not perform that well and had a high level of no calls. And also for some of the labs, we do see a little bit higher level of error rate, and the errors in these two examples are at least dropping out, which also are not unexpected when it comes to low level of DNA input. However, what we wanted to show is that even though we did not get full profiles, complete profiles for many of the different attempts, it's still useful and usable to obtain information downstreams due to the fact that there are a high number of markers in total included in the panel. So even though you have a 50% marker dropout, so you only have SNP calls for 50% of the markers, you do have calls for 2,500-ish SNPs, and the information for these number can still be useful. And we just wanted to show an example of this for the Y chromosomal SNP. So for the good quality samples, and this is the result from a representative sample, we did see that when you have a high level of input DNA, you do call most of the Y SNPs, in this case around 800. And as expected, when you decrease the level of, or the amount of input DNA, the number of called Y markers are also decreasing. However, in this example, what we do see that even though the number of SNP markers are decreasing, they're still yielding the same level of resolutions when, in this example, comes to haplogroup interpretation. And the same applies to kinship analysis, having a partial profile. So this is a simulated example. So going for a third-degree relationship, which is corresponding to a first cousin relationship, or a great-grandchild relationship. And if you have a complete profile representing the dark green here and the dark red here when it's unrelated, you do see quite a high level of information obtained using the kinship SNPs. But what we do see also, if you only have 25% of the kinship SNPs typed, meaning the majority is untyped, you still can get a high resolution, be able to, in principle, separate all related third-degree relationship compared to unrelatedness. So still, even though you may have a lot of dropout, marker dropout, the kinship prediction may work fine with partial data. So then we come to the second part of the interlab study, and we've focused here on interpretation, meaning that a different application when you have your SNP genotypes and how to make use of them for various applications like skin color, eye color, hair predictions, or kinship analysis, etc. So here we wanted to exercise this for the same number of labs that participated in the lab part, the part 1 of this interlab study. So each lab got the same case question and case scenarios and the same SNP genotype data, and were tasked to provide an answer and do the inference based on statistical tools or other tools and reference data for downstream interpretation. So the participating lab got the different tasks for 3 different tasks. One was that they wanted to, they were tasked to interpret whether 2 individuals were first cousin ones removed or unrelated to each other. So all labs did get the same SNP data and the same allele frequencies for these SNPs, and that should calculate the likelihood ratio for these 2 hypotheses. The other case scenario was to mimic a DVI scenario where each lab got 4 SNP profiles, also included partial SNP profiles from 4 bodies that were sort of found outside and were believed to be from individuals that disappeared 50, 60 years ago. They also got reference samples from family members to all of these missing individuals, and the task was to try to establish the identity for these 4 bodies that were recovered based on the SNP profiles. And the third case that were given was to do ancestry and phenotypic prediction based on SNP profiles from crime scene samples from unknown individuals. So they got the SNP data and also reference data and tools to predict eye color, hair color, and skin color, and biographical ancestry. And since there are a number of different tools and reference data available to do this, but there are also some of these may be complex. So we wanted to provide different labs with a tool if they did not have any tool of their own. So FamLink 2 were sort of improved in the sense that a number of new features and functions were developed and implemented. And it was done by Daniel Kling. It worked a lot on this, which also was provided along with the SNP data and the case questions and with reference data. So the majority of the participating labs did use the FamLink 2 to infer and calculate the statistics for different exercises. However, also other software and tools were used. So the results show from each lab that participated in this part that overall there was a large concordance between the labs when it comes to the statistical analysis and the outcome. We did, however, see a little variation when it comes to how they verbally expressed and interpreted the sort of the conclusions from these different case scenarios. So with that, I would like to sum, and we believe that there is a need for standardization of forensic SNP markers in order to improve and make it more easy to use SNPs in forensics, because SNPs in forensics are useful and has been shown to give information in many different cases. And the force panel is a set of around 5,000 SNP markers for various sort of different forensic applications that can be used as a set of such a standardized marker set. However, there are other similar panels available as well, like the Intelligence Kit from Kaigen. And what we wanted to test in this interlab study that I presented here was to see if the same set of SNPs can sort of be applied with different assays and different sequencing strategies in order to obtain useful SNP data. And to conclude, what we showed in this interlab study, that the different assays that were included shows that it is possible to customize the complete workflow going from DNA to genotype. And we will, in the upcoming paper, also discuss which of the different parameters that the different labs uses may have the highest impact on the ability to obtain good quality SNP profiles. So of course, a study like this, a study setup like this, can only be done through collaboration. So once again, we would like to thank all the collaborating labs and individuals that participated in the study. It's extremely useful, and more details will be available when our paper about this study comes within a couple of months.